Hello, people. Welcome to Cell Biology. I'm glad y'all made it. Or some of you.
You're late, Slish.
What's that? Is that
You're
Bailey?
late.
I'm late.
No, that's Rachel.
I am late. I'm 3 minutes late because I was trying to get a quiz together for you guys. And I did that. So what'd you say? We just go do that quiz. Let me show you where it is. You probably know, but in case anybody comes late, they'll know what we're doing. Here it is. Bank. April 4th. No, April 1st. All right, go ahead and do that. I will be back at 11:15. Okay, and we are back. Let's take a look at this quiz. So I don't know if you noticed, but the second question was worth twice as much as the first question because it was a much longer question. Where did I get here? Boom. And oh, I hit back to the course. Okay, and a quiz. Okay, holy shit. Sorry, I just realized I gave you the same question twice in different ways. Oh well. I guess I should put these quizzes together before the last minute. Oh well. Growth factor binds to RTKs to transphosphorylate. This makes a.
We can't see the quiz.
Oh shit! I do that every time. Thank you for telling me. Okay, that makes an SH2 domain that attracts PI3 kinase. This enzyme phosphorylates. Oh, look at this. They're all in order for me. I don't even have to think. Ta-da! Okay, now the next question will be no more difficult. Oh, come on, man. Screen share. Share. Look at this, the PI3 kinase mechanism. Where have I heard this before? A blank binds to receptors, typically a growth factor, which comes together in phosphorylate, creating SH3 domains, right? Wrong. SH2 domains. PI3 kinase is recruited to the receptors and phosphorylates phospholipid PIP2 into PIP3, tracks proteins with the pH domain, which includes, I believe, AKT. And kinase phosphorylates beta-catenin, blocking beta-catenin, TCLF, and there you go. Okay. So hopefully y'all got that right since it was pretty friggin' easy. But you know, little bonus for people who made the effort to show up today. Okay, so speaking of today, what we're going to do today is start the second half of this exam material, which is going to be Xenopus laevis early development. So I really like this. I wish it was in the chapter. It is, or in the book. It's not. What I really like about this is that it shows you how these hormones, what they do, and why they're important in early development. So you know, it's kind of like throughout chapter 23, it's all kind of esoteric. You know, it's like this happens. Where does it happen? Don't worry, it's different in every cell. Blah blah blah. That's kind of what cell biology is like. So this can be this kind of like connects all of that cell division stuff. I mean, all the signal transduction stuff to it. Something that is actually doing something in the organism. So we'll talk about how early development happens up to the point of gastrulation. And then we'll talk about what hormones are responsible for all this happening and how the signal transduction of those hormones causes changes in the cell that then make the development happen. So if you're concurrently taking developmental biology with me, this is basically an abbreviation of both exam 1 and exam 2. So this is going to be really easy for you. But for the rest of you, it should be like dramatically exciting. I mean, don't bother putting socks on during this lecture because I'm going to knock them off. It's going to be awesome. So without further ado, Xenopus early development. By the way, because it's not in your book, I did more than just write notes for this section. I also have have it all written out in text form as well. So hopefully that's going to help you with this. And you each owe me $5 for that text version. Okay, so just send it to me. I do Venmo, PayPal, whatever. You know, it's funny, I joke about that, but there was a professor not that long ago in communications who charged his students like $10 a piece for something, like something about the syllabus or something. He said it was extra, so they had to pay him directly $10 for it. I'm like, what?
Ew.
Pretty scummy. Although I will take money if you want to give it to me. I'm just not making you give it to me. Okay, early development, gastrulation. It's all about axes of symmetry, the axes of symmetry. And what we mean by that is axes around which you've got sameness, whatever. So if you look at different organisms, like animals, we're only talking about animals here. Look at a sponge, doesn't have any axis of symmetry. It's just, let's see, I could come up with this. Thank you, Christy, for not coming up with a smart ass comment. Thank you. Although I know you could. Okay, sponge. So sponge has got really no axis of symmetry. It's kind of different from anywhere you look at it. And these cells kind of build off of each other and form a rough structure. And same thing with coral. They're going off in all different directions. And so there's really no axis of symmetry. When we get to the next level of animal structure, you talk about a one axis of symmetry kind of an animal. So here's your, I think this is a sea anemone, although I'm not a sea person. Not a fan of the ocean in general, if you don't mind my saying. So you've got a kind of a circular thing. You've got one axis, which is top and bottom, okay? And then other than that, it's radially symmetric. As you go around, it's all the same all the way around. So that's one axis of symmetry. And then here's some more examples of one axis of symmetry. Our friend, the jellyfish, who makes swimming in the ocean so much fun. This is another sea creature. And this is a starfish. Sometimes I can BS my way through things and sometimes I cannot. So here's something I know more about. This is the brown trout, favorite of fly fishermen. So the trout then is a chordate and it is a vertebrate. And as such, it's got three axes of symmetry. So you've got dorsal, ventral, and I always remember dorsal by the fact that fish have got a dorsal fin, right? So this is the dorsal side and the belly is the ventral side. Then it's got anterior, posterior, front and back. And then you've just got your left and right after that, okay? So if you go from the middle of the fish that way, it's kind of the same as if you went from the middle of the fish to this way. For the most part, there's some changes in the organs and stuff. And so that is your three axes of symmetry. And here are some animals that have that. This is an invertebrate, but it's got, it's a chordate though. It's got a spinal cord. So it's got three axes of symmetry. This guy has got three axes of symmetry. This gentleman, I don't know. They could all be female. What the hell do I know? This is a male though. This is a male. And he's got three axes of symmetry as well. Okay. So that's my introduction to axes of symmetry. So Xenopus laevis. So this is the Xenopus. I used to keep these at Plattsburgh State, and I worked with them when I was in doing my postdoc. These are dumb. These are dumb animals, and they'll eat anything that comes near. We used to do these developmental studies in my postdoc, and we'd start with like maybe a whole bunch of fertilized eggs, and then we'd get maybe a dozen or 2 dozen tadpoles and put them in a bowl. And the tadpoles would turn into frogs, and you'd have 2 dozen frogs. And then you look a couple of days later, and well, now we only have a dozen frogs. Where do the other ones go? And then a couple of days later, you would have 3 big frogs, and then a couple of days later, you'd have 1 big frog. They'll eat anything. They eat each other as fast as they eat anything else. Amazing animals. And that's why you should never release one of these into a pond, because they'll just eat everything in the pond. They'll all grow everything. Okay, anyway, back to work. So this is a mature egg cell. They start out as oocytes and then mature to the point of egg cells. Now these cells are halfway through meiosis. So they've gone through one round of meiosis and they've got a polar body attached to them, which I don't have here. When the sperm enters, they'll do that second round of meiosis, and then the two nuclei will fuse and you'll have a zygote. The thing about the Xenobus Xenobus egg is it's brown on top and yellow on the bottom. And I mean, top and bottom, it's kind of relative, but the brown part is the animal pole and the yellow part will be the vegetal pole. The yellow part's yellow because it's got a lot of yolk in it. And while the egg is being made, you get specific messenger RNAs that will be attached to the top of the pole, at the top of the egg and at the bottom of the egg. And that will make this a certain tissue and this a different tissue once it starts to divide. And an important thing to keep in mind is that's really going to drive development until you get to about stage 12, because you'll get no transcription from embryonic nuclei until you've gone through, not stage 12, 12 divisions. So once you've gone through 12 divisions, then the nuclei will turn on in the zygote. But early development's all driven by maternally laid down messenger RNAs. Okay, so the first thing that happens to start this whole process is Mr. Sperm comes along and enters. So the sperm will always come into the animal pole at some point. And when it does enter, it activates IP3 production and release of calcium, and that flashes all the way around the egg really quickly and shuts the egg off so it can't accept another sperm anymore. So that avoids polyspermy, which would result in extra nuclei and would mess up the embryo. So the first thing that happens is sperm enters, you get this release of calcium all around, and boom, now it's going to be a zygote. It'll go through that second round of meiosis, and then you end up with these two polar bodies up here, which just kind of go away. I'm not worried about those. But important thing to remember, and we'll talk about more later, is where that sperm enters will be the ventral side of the embryo eventually, and the opposite point then will be the dorsal side. And the dorsal side is the key. That's what's going to organize the whole body plan. So opposite of sperm entry becomes the organizer. Okay, so sperm enters, cell divides into two. Now, if you took this early zygote, split those two cells apart, you'd get two identical frogs, okay? They would be clones of each other because they'd be genetically identical. And they'd come out, you know, maybe a little smaller than a regular frog, but, you know, one more cell division, they're just equal to them. So at this point, those two cells are identical. Now the second cleavage comes across this way, and now those four cells are not identical. We'll see later that there are some dorsal determinants that are laid down opposite of the sperm entry point. So if you take these two cells out, they would become like almost an embryo, but only have dorsal structures. Take these two embryos out, they would be just kind of the belly of a frog, just some ventral tissue, kind of not much of anything to talk about there. So anyway, at this point, you no longer have four identical cells. And then your next cleavage will be across the middle here, cutting off the animal pole from the vegetal pole. And that now turns this zygote into two different tissues. So at this point, when the sperm enters, this cell is totipotent. It can become anything. Right, it has the ability to become anything that a frog needs to be a frog. At this stage, the two-cell stage, both of those cells are totipotent. They can do anything it takes to become a frog. Now, at this stage, each cell is pluripotent. Okay, it become many, many different things, but not everything. So each of those cells is no longer totipotent. They're now pluripotent. And every step after this, each cell gets a little less potent until you get to fully differentiated cells, and then they are just what they are. They're no longer capable of dividing. But anyway, once you get to this stage, the top cells now are going to be one tissue, which if you took them off that embryo, they would become skin. They'd become skin tissue. And the bottom then is this yolky endoderm stuff, and they would just become kind of non-differentiated yolky endoderm. So anyway, that's important. That cleavage gives us two different tissues, ectoderm and endoderm. And of course, it will keep on dividing and dividing and dividing until you get a whole lot of cells in one ball. Now, let's watch this happen because I think this is really worth seeing.
The fertilized ovum undergoes rapid divisions and forms a blastule. Each division normally takes about 30 minutes. The blastula develops into a gastrule. Neural folds develop and fuse to form the spinal cord and brain.
Pretty.
Eventually, the embryos hatch as free-swimming tentacles or larvae. The tentacles undergo metamorphosis and tiny frogs emerge that eventually grow into adults.
Okay, so there was one point there I was going to back up to, but it's too late already. If you remember early on, you had cells, and suddenly you saw cells spreading all over. That's epibole. We'll talk about that shortly. But anyway, blastula. So divides, and after it divides enough, you get this blastula stage, and the blastula is signified by the fact that you've got this big fluid-filled area in the middle called the blastocele. So it's open here in the middle. And then you've got your ectoderm on top and your endoderm on bottom. You'll notice that the ectoderm cells are smaller. Ectodermal cells are going to divide faster than endodermal cells. These vegetal cells are going to be really, really big, and these are tiny. And you can see here's bigger cells, and then they keep dividing till you get tiny cells. Okay, so you've got ectoderm on top, endoderm on bottom. Now, ectoderm is fated to become either skin or nerve tissue. Fated means eventually this is what it's going to become, which means there are certain genes turned on in them or certain messenger RNAs in there that have already pushed them a little farther down that differentiation pathway that now they have to go forward from here. So differentiation involves a whole lot of changes from one cell type to another, and it's pretty much irreversible for the most part. So every time you get to a decision point and change, you've changed your fate, and then you have to keep going down that pathway. So these ectodermal cells then are fated to become either skin or nerve tissue. Endoderm then is fated to become gut, basically, your GI tract. So that's about all that's ever going to be. So you're still missing a whole lot of stuff that makes an animal. You're missing muscle, bone, blood, reproductive tract, kidneys. All that stuff is going to come out of a third tissue, but we don't really have it yet. What's going to happen is right here at in the marginal zone between the endoderm and the ectoderm, the endoderm will release some hormones, and those hormones are going to activate signal transduction pathways, TGF-beta like, and that will turn these ectodermal cells into a new tissue called mesoderm. So this whole marginal area then becomes mesoderm. And mesoderm is going to give us our muscle and bone and blood and other things as well, but that's the main stuff. Okay, so now we've got all three different types of tissue that we need to form an embryo, but they're not ordered correctly, and that's what gastrulation is going to do for us. It's going to order those tissues. So at this point, we still have one axis of symmetry, top and bottom, right? But from any point in the center, it looks the same all the way around. So what did I want to say here? So this is kind of like a mid-blastula stage. Okay, so you've got animal cells on top, and we do experiments where we cut these off. We call that animal cap. And if we cut that off now and let it just grow in tissue culture, it will become skin. It'll become skin. If we cut the middle part out and let it grow, it will become ventral mesoderm, which is basically blood and blood vessels. And here's your endoderm, which is just undifferentiated usually when you take it out. But those are our three different tissues. Now, how are we going to get an embryo from this? I'm glad you asked. So here we're getting to that stage again. Here's your blastula, huge vegetal cells, smaller ectodermal cells. And now here we're at the late blastula stage. Now what's going to happen here at the late blastula stage is these ectodermal cells are going to divide really fast. So you're getting a lot of small cells. And what happens is some of them are going to stay here, but some of them are going to spread down around the embryo from in all directions in 360 degrees, all the way, around to meet this point that's opposite of where the sperm entered. That process of those cells covering the embryo is called epiboly. Some people say epiboly, but E-P-I-B-O-L-Y, epiboly. And that's going to put skin on the outside of your embryo. And let me tell you, you don't want to live without skin. Skin's pretty important. It's keeping everything in. So that's epiboly, and that's going to be happening at the same time as gastrulation happens. So what is gastrulation? What happens in gastrulation is, hang on a second, let me see if I better. Yeah, let me hang on here for a second. Go forward. That slide there. Okay, so here we are at late blastula. You've got mesoderm and endoderm and ectoderm. Ectoderm is going to cover the outside. Now over here, opposite of where the sperm came in. So the sperm came in here. Opposite here, we're going to form this dorsal lip. Dorsal, because this will be the dorsal part of the frog in the end. And you've got a blastopore here with the dorsal lip. So what's going to happen now is as these cells are dividing, these mesodermal cells are going to come inside and then crawl up that way. So we'll have both mesodermal and endodermal. Oops. Damn it. Just blow it up. So what's happening here, backing up one, is you form this blastopore lip, and when you do, mesodermal cells and endodermal cells will crawl inside the embryo and across the top of this blastopore, or blastocele. So here they are coming in, and they're dividing, so you're making more of them as you're doing this, and they're coming in across the top of the embryo like this. Now there's a space between the endodermal cells that are involuting and the endodermal cells that are just sitting there. and that space is called the archenteron. That will be the inside of your gut in the end. So this will be the anus over here, and the mouth will emerge over here much later in development. So the archenteron is kind of the primitive gut. So you've got mesoderm and endoderm coming in this way, and at the same time, ectoderm spreading around the outside, and the ectoderm will meet at the blastopore over here. So here you can see this ectoderm has spread all this way, and it's meeting over here. You've got this yolk plug that will go away. Here's your archenteron, and the blastocele now has been pushed down out of the way, and it will disappear eventually. That fluid will just be pumped out of the embryo, and your center will be archenteron. So what we've done here with gastrulation now is given us three layers of tissue, ectoderm, mesoderm, and endoderm on top of each other. And it's also given us the three axes that we need. Over here, where the archenteron's coming out, that will be posterior, right? That's the butt. Butt's coming out here. This over here will be anterior. The top here is dorsal. The bottom is ventral. And now into and out of the screen is going to be left and right and left, whatever. Okay, so that's what gastrulation is doing for us, is giving us those three axes. And let's see. Here's how it starts. Here's where the blastopore lip is going to form. These little bottle cells form from the endoderm, and the leading edge then, which is mesoderm, will push in because of those bottle cells pushing it in. You can imagine these cells are dividing, and as they divide, cells push down, and then these bottle cells make them go in instead of straight down. And then they go up this way, and now you've got mesodermal cells and endodermal cells moving in to the blastocele and going right up along the ectodermal cells that are there. And so here we are at our gastrula. You've got endodermal cells here, endodermal cells down here, gut in the middle of it. You've got mesoderm, which is going to be skeleton, circulatory system, muscles, excretory system, reproductive tract, that's here. And then outside, you've got ectoderm, which will be like, we just saw epibole happen and ectoderm went all over it. So that's your skin all over, but also the ectoderm that's right over, most adjacent to this mesoderm, will be induced to become the central nervous system. So you'll get a brain over here and a spinal cord all the way down to here. Okay, and that's the next thing. Let's see what this is all about. I don't remember what I put here.
Cross-section of an embryo of the frog's anthers, we can see that at this point it is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called epibole. As gastrulation proceeds, a cavity called the archenteron forms while the blastocele progressively shrinks. The archenteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the endoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm towards the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined. The endoderm gives rise to the digestive and respiratory tracts and associated structures. The mesoderm gives rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. The ectoderm gives rise to the skin, sense organs, and nervous system.
Ta-da! All right, let's watch one more of these. I've got another video by a woman who does a really awesome job. I hate to say it, but better than me. Hang on a second. Shut up, you. Okay. Can you guys see this woman holding an embryo thing? Yeah, okay, good. She's pretty awesome, by the way.
Inside the archenteron. Okay. So here is a fully formed gastrulum with a so the animal cells have completely come around to migrate inward. And this edge also, again, this would be the dorsal lip where the migration first began to involute to form the archenteron. This side here, where the cells are on the other side of the what's left of the vegetal wall, that is the yolk plug. So this side here, where the cells are involuting, also is the ventral lip. So this opening that the yolk plug is filling is called the blastopore. Inside the archenteron here, all of the yolk cells are inside the archenteron. The lining of the archenteron is the endoderm, the yellow cell layer. The outer blue layer is the ectoderm, and the middle layer is the mesoderm. And that's it.
And that's it. Okay. Okay, so just to finish, Pibli then is the expansion of that outer sheet of cells because they're dividing so fast they go this way. Involution is when they come in. And here is a cross section through the late gastrula, yolk plug, dorsal lip, ventral lip, archenteron. Okay. So that's what I wanted to cover today. And then next Tuesday, I will finish. We'll talk about the inductions, the signal transduction happening and things like that, and how that causes changes. And then Thursday, I'll do a quick review. And then Tuesday following is our exam, correct? Yes, Dr. Slish. Pinello, I got an up sign from. Good. How's everybody else doing? Everybody happy? That's about all I have for today. I'm getting some thumbs up. Tyler's my man. Thank you, Tyler. I got another thing for you. I'll be emailing you today. Okay, then. The other thing is I'm going to be uploading a game soon. Tyler helped me finish off the game. It's all set to go. I just need to upload it, and then I'll put the link into your Moodle page. So that game 3 is ready to go. Okay, guys, thank you so much for coming. I hope we can do this again sometime soon. I enjoy your company so much, and talk to you again.